Hi everyone, this is Alexis Murillo. I am a postdoctoral researcher at the Comprehensive Center for Precision Oncology at the Universidad de São Paulo, Brazil. Today, my colleague Cristóbal Cerro Trucoso from the Center of Human Genetics in Belgium and I will moderate this edition of SNEV Characterization Series. On behalf of the SNEV board, I would like to welcome you and thank your presence for this talk presented by Dr. Juan Pablo Tosar. Juan Pablo Tosar is an assistant professor and head of the Analytical Biochemistry Unit of Universidad de la República and senior researcher for the Functional Genomics Laboratory and Institute Pasteur, both institutions located in Montevideo, Uruguay. His research is based on RNA biology with a focus on extracellular vesicles, and Juan Pablo Tosar also participated as a reviewer for several international grants, fellowships, and scientific publications. Today, Dr. Tosar will share with us his experience through the talk entitled Things Are Not Always What They Seem, Characterization of Vesicular and Nonvesicular extracellular RNAs. Please remind that you can put your questions on the chat box during the presentation, and they will respond in the end. And also in the question section, you could open your mic to ask Dr. Tosar directly. So welcome, Dr. Tosar. It's a high pleasure to us having you here. Thank you for accepting our invitation. Please feel free to start your presentation. The word is yours. Thank you.
Thank you, Alexis, for this kind of invitation. Thank you, Alexis, Cristobal, and people at SNEV for inviting me to this talk. So I hope we will have the chance just to discuss about vesicular and non-vesicular extracellular RNA. I know these series are about the characterization of extracellular vesicles, but I will try to make the point that we also need to understand what's outside vesicles to understand what is inside. And this will not be a review of the field because it's quite large. It will just be a talk trying to provoke you in order to see whether as a community we can think in other directions and giving a few examples on that. So with this, I will move to this diagram. This is extracted from the webpage of a vendor, but you can find similar diagrams in many reviews about RNA-mediated intercellular communication, which we could summarize as the hypothesis of exosomal shuttling of microRNAs, where an exosome-generating cell, a donor cell, releases extracellular vesicles containing microRNAs, and these vesicles are internalized by recipient cells, either by fusion or by endocytosis, but somehow these microRNAs get into the cytosol of the recipient cells where they regulate gene expression by targeting miRNAs. And I'm not implying that this model is not true, but I think we can challenge it a little bit to see whether we can also consider other hypotheses. So this is how we typically think about intercellular communication. So we have a shipwreck and we have this person alone in this island. This will be the donor cell. And the donor cell wants to send a message to a ship or to land, and the message needs to make sense, like, for example, help me. And that message could be encoded in RNA. But because the extracellular space is full of RNases that will degrade the RNA, we typically think that this message needs to be encapsulated inside a carrier, and extracellular vesicles are typical carriers for this message. So this is actually a model, a mental way on how we think about intercellular communication. And these models have several assumptions. For example, we need to assume or show that EVs are the main carriers of RNA in the extracellular space. We tend to think that outside EVs, the RNA is degraded fast and cannot exist. We also usually assume that microRNAs are abundant inside EVs. So they are abundant message. That cells select specific microRNAs for release into EVs, presumably to communicate a message that, let's say, for example, that microRNA 142, which has invented one, will be decoded in the recipient cell as a help signal. And we also assume that one cells internalize the extracellular vesicles containing the microRNAs. These microRNAs will be able to target specific mRNAs to regulate gene expression. So let's go one by one these assumptions and see what can we say about them. So let's start with the first, that EVs are the main carriers of RNA in the extracellular space. So in 2015, we did this very simple study where we grew human cells in serum-free cell condition medium, and this is quite important. And we purified large EVs and small EVs by ultracentrifugation. So we have a 16,000 G pellet corresponding to large EVs, a 100,000 G pellet. And then we had the 100,000 G supernatant, which we said this is our non-vesicular fraction. And we did small RNA-seq in each fraction, but before that, we just quantified the RNA. And as you can see, most of the RNA in the extracellular space was not associated with the vesicular pellets, which is this bar in red and green, but actually remained in the non-vesicular, in the 100,000 G supernatant. And when we did small RNA sequencing, most of the reads in the extracellular fractions, but especially in the non-vesicular extracellular fraction, corresponded to tRNA fragments. More than 60% of the reads in the non-vesicular fraction were tRNA fragments. And that was somehow strange. It was not simply a question of quantity, it was also a question of qualitative. There were qualitative differences. Inside the cell, we could sequence tRNA fragments coming from different tRNAs, and they had different sizes. Outside the cells, and especially outside vesicles, they were mostly 5' tRNA halves of tRNA glycine or tRNA glutamic acid, and their size was precisely 30 or 31 nucleotides. So we asked, what protects these tRNA halves from degradation? And then we realized that these tRNA halves are self-complementary, and we showed in vitro that they can form dimers. And these dimers have this predicted structure, which is a double-stranded RNA structure, and extracellular ribonucleases tend to cleave single-stranded RNA much better than double-stranded RNA. So this will explain the increased stability of these tRNA halves. So we asked what will happen if we inhibit extracellular degradation. So we added RI for ribonuclease inhibitors to the media, and by Northern blot, instead of seeing these 5' tRNA halves that we sequenced, now we were observing full-length tRNAs. If we inhibit the degradation, now we see full-length tRNAs, implying that cells are not releasing the extracellular tRNA halves. They are releasing tRNAs, and the tRNAs are being cleaved in the extracellular space to produce these tRNA halves. And this observation was independently confirmed almost simultaneously by a group at Cold Spring Harbor in New York. In this case, they didn't use ribonuclease inhibitor. They did a knockout for an RNase in a cell, and they observed exactly the same. So the title of this talk is Things Are Not Always What They Seem. And here we have the first example. So I mentioned that we sequence small RNAs outside cells and inside cells, and outside cells, we observe a huge enrichment of specific tRNA halves, represented here by red and blue. So these tRNA halves in red and blue outside vesicles, outside cells, are much more enriched than inside cells. And we could interpret this as an example of selective release. So the cell somehow looks for these red and blue RNAs and releases them into extracellular space. That was our initial interpretation. But it turned out that what's happening is that cells are releasing full-length tRNAs, probably non-selectively. These full-length tRNAs are being degraded by extracellular RNases to produce tRNA halves. And some tRNA halves are more stable than others, and they accumulate. So non-selective release coupled to differential extracellular stability is undistinguishable from selective release. So we need to be careful on how we make interpretations, we make hypotheses on what's going on in the biology by just seeing a snapshot by sequencing, because there's a lot of survivorship bias going on. And we will actually show this by inhibiting RNA degradation, as I mentioned. So the second assumption in this black box is that outside EVs, RNA is degraded fast. Actually, today, we accept that there are other potential RNA carriers in the extracellular space beyond EVs. So we have large EVs and small EVs, but actually there are ribonucleoprotein complexes and lipoproteins. They all could transport RNA in the extracellular space. But what we usually do not consider is the possibility of having naked RNA in the extracellular space. So we accept the possibility of having naked RNA inside EVs. Like for example, in this painting, this shell would be like an EVs, but as soon as Venus is going out of this shell, her nakedness needs to be covered, because we assume that extracellular RNAs will regrade immediately any RNA. So we actually tested that. So we purified RNA from cells and incubated the purified RNA in human biofluids, urine, serum, and cerebrospinal fluid for different periods of time. And as you can see, the RNA is rapidly degraded, but there are some species that remain, especially in CSF. And then by Northern blot, we try to identify which species were these. So ribosomal RNAs, or this 7SL, which is an RNA Pol III transcript, they were degraded very fast, in less than a minute. The same happened for example, the full-length glycine tRNA that you can see here. But you can see again that the incubation of this full-length tRNA in biofluids produces tRNA hubs, and these tRNA hubs are very stable. And what's interesting is that this depends on which tRNA we are looking for. If we are looking for tRNA lysine, which has a lot of base modifications in the anticodon, it is much more resistant to degradation. So here we have an example of a naked RNA that is intrinsically resistant to degradation in human biofluids. So tRNA lysine is more resistant than other tRNAs and other ribosomal RNAs. But tRNA hubs produced by the extracellular fragmentation of full-length tRNAs, they are extremely stable as naked RNA. Their half-life, measured by these assays, is more than 2 hours in CSF at 37 degrees. But how can we explain this extreme stability of these species? We offered an hypothesis or an explanation earlier, the fact that they could form dimers, but that's true for glycine tRNAs, tRNA hubs. It's not true for any tRNA hub. And actually, by doing these assays, we were observing that different tRNA hubs were actually very stable in biofluids. So then we made this question, and I will go to EVs in briefly, but please follow me in this non-vesicular, in trying to understand what's going on with the non-vesicular RNA. We tend to think that when a full-length tRNA encounters an extracellular ribonuclease, these are ribonucleases we clip at the anticodon producing 2 tRNA hubs. But these tRNA hubs are single-stranded RNAs. This is single-stranded RNA, and it will be degraded fast. So maybe what the RNases do is they just clipped a phosphodiester bond at the anticodon loop of the tRNA, but now we still have, instead of a full-length tRNA, now we have a nicked tRNA, but it's still a double-stranded thing, and this will be stable. But how, if this species really exists, how can we study them? Because most RNA extraction kits, small RNA sequencing, and Northern blot, they all involve RNA denaturation steps. So if these RNA species actually exist, they will be unfolded, they will be split by our RNA extraction or analysis methods. So we decided to reproduce this in vitro. So here by Northern blot, we can see a full-length tRNA. This is tRNA glycine. We treat this with RNase 1 for an hour. This is the RNase which is more abundant in human serum. And as you can see, we see a complete conversion of the full-length tRNAs into 5' tRNA hubs. And then we reasoned that if these nicked tRNAs were producing these tRNA hubs, because this is a Northern blot, here we are running the RNA on a denaturing gel. So we're splitting any double-stranded RNA species that we have. We could repair this by using a sequential ligation, first with T4PNK to repair the extremes, and then T4 RNA ligase 1. And when we use both enzymes sequentially, actually, we recovered a full-length tRNA again. But you could think, oh, but maybe we are ligating in trans. So we are ligating a 5' tRNA hub with a 3' tRNA hubs, but they were not together. We're just doing an artifact. But that's not the case, because if we hit the RNA before this ligation protocol, this doesn't work. This means that this is a product of ligation in cis and actually proves the existence of these nicked tRNAs. And to show this in a different way, we did native gels before a Northern blot. So this is no longer a denaturing gel. And here, if we treat the RNA with RNase 1 for an hour, we no longer see it running as a tRNA hub. We see it running as an almost full-length tRNA. But if we purify the RNA with trisol, with miRNA or any kit containing phenol, or if we hit the RNA, now it runs as a 5' tRNA hub. This means that these nicked tRNAs really exist in the extracellular space, and because of how we analyze them, we are forcing their separation. And it turns out that these tRNAs are extremely stable to degradation. The tRNA halves are not. We show this by treating the RNA with RNase 1. Then we purified these tRNAs. We treat it with RNase 1 again, and they resist. But if we hit them and we treat them with RNase 1 again, then they are gone. So this actually explains that probably what's going on is that cells are releasing full-length tRNAs. These full-length tRNAs are being clipped into tRNAs, and that's it. These tRNAs are the stable entities in the extracellular space, not the tRNA halves. What if we want to sequence these tRNAs? Either we use an RNA extraction protocol that involves the denaturation of them, so we will sequence them as fragments, or otherwise we cannot sequence them unless we repair the nick. Why? Because reverse transcriptases, they will go from the 3' end to the 5' end of the tRNA, but as soon as they encounter the discontinuity in the anticodon loop, because of the clipped phosphodiester bond, they will not be able to reverse transcribe the molecule. So we need to repair first and then do RT, PCR, and sequencing. So why I took some time to tell you this story, because it's another example that things are not always what they seem. You will remember the Greek philosopher Plato. So Plato actually presented his allegory of the cave, where we have some prisoners who were born in a cave. They have a wall behind them, and they are only allowed to see the wall which is in front of them. And they see the shadows of objects that are moving behind them. And because they were born in this cave, they can think that these objects are the reality, are the real thing, that they live in a 2D reality. And actually, that's not the case. Those are only projections. And the same happens with sequencing, especially with small RNA sequencing, that we are only seeing with sequencing a one-dimensional projection of a 3D or a 4D, because it's actually dynamic reality. RNA structures live in 3 dimensions, and sequencing cannot tell us that. And if we don't take this into consideration, we can lose, our interpretations can be incorrect. And this is important because also the type of sequencing method that we choose will completely change the output, and it can change our conclusions. For example, let's say that cells are releasing tRNAs. These tRNAs are being clipped in the extracellular space into tRNAs. These tRNAs then denatured spontaneously into tRNA halves, and some of these tRNA halves can self-associate to produce dimers. What will happen if we sequence all these heterogeneous populations? So all these are very different molecules from a chemical perspective and possibly from a biological perspective as well. If we use standard small RNA sequencing, like microRNA sequencing, we cannot sequence full-length tRNAs. We cannot sequence the 3' halves of tRNAs because they typically contain modified bases that this sequencing, that the reverse transcriptases that we normally use cannot handle. So we will only see 5' tRNA halves in all cases, but this is a ground truth, okay? So this is very different. We can use more refined methods like TigerSeq, which can sequence full-length tRNAs well, but it will interpret nick tRNAs probably as a collection of tRNA fragments. And then we can do other methods specifically designed for the sequencing of nick tRNAs. But what's important here is that all sequencing methods will be biased, and we need to understand their bias, and we need to use the bias in our favor as an enrichment strategy without thinking that what we are seeing by sequencing is a reality. We are losing a lot of things. We are losing a lot of information, and we need to bear always that in mind. So I will try to also illustrate this point about fragments versus full-length things with this example. Here we collected conditioned medium from U2OS cells. So these are a human cell line. The cells were grown in DMM plus 10% beta-bovine serum, so standard conditions for different periods of time. And here we're just collecting the media and analyzing it by northern blot using a probe against the 5' tRNA glycine. And as you can see, we see that with time, we start seeing a collection of full-length tRNAs and tRNA halves. So both things, the full-length tRNAs and the tRNA halves are in the cell conditioned medium. Also 7SL, which is an RNA pol3 transcript. But then we took this cell conditioned medium and we loaded it on the bottom of an optical gradient, and we centrifuged that overnight. So we actually did a flotation gradient where the EVs will float because of their lower density. And this protocol actually separates pretty well the non-vesicular components from the vesicular, from the EVs that will float and will appear in the lower fractions. So fractions 12, 11, 12, 9, 8 actually contain non-vesicular RNA. You can see that by BSA. This is the albumin from the FBS. You can see the ribosomal proteins because we also have ribosomes there. So actually this paper was showed the existence of these non-vesicular ribosomes. But here by northern blot, we can see the tRNA halves. We are not seeing full-length tRNAs in the ribonucleoprotein in the non-vesicular fraction. And these tRNA halves actually, we know now they correspond to unique tRNAs. If we go to fractions 3 and 4, which contain CD9 and CD81, so here we have our stringently purified EVs. Here we see by northern blot full-length tRNAs, almost no tRNA fragments. So without separation, here in the left, we see both full-length tRNAs and tRNA fragments, but actually the full-length tRNAs are in the Vs, the tRNA fragments, the unique tRNAs are outside the Vs. And this is quite important, I think, because these tRNA-derived fragments in EVs, we earlier sequenced them, we earlier observed them. Are they really present inside EVs, or are they a consequence of contamination with non-vesicular unique tRNAs? These non-vesicular unique tRNAs are highly abundant. So if we don't use a very stringent separation method, they will contaminate our EV preps. Actually, this is a recent preprint from Ken Whitworth's lab, where they managed to use a single vesicle analysis methods, and using hybridization proofs, they kind of managed to show that some of these tRNA fragments might be inside EVs. So I'm not saying that they are not, but if you look at the northern blot, most of the signal actually corresponds to full-length tRNAs in the EVs, not tRNA fragments. So, as I mentioned earlier, unless EVs are stringently purified, abundant non-vesicular RNAs will contaminate EV preps. And we should note that several non-vesicular RNAs are already resistant to RNases. That's why they are there, because if they were not resistant, they wouldn't be there if we are growing cells in the presence of FBS, which is a huge source of RNases. So simply doing an RNase treatment does not discard non-vesicular RNA contamination. If we do a combination between detergent treatment and RNase treatment, then it could make more sense. But simply showing that an RNA is RNase resistant is not enough to prove that it's inside EVs, in my opinion, at least. Now, changing a little bit the focus, let me say that it's hard to find what you're not looking for. And this is important for sequencing studies. I don't know if you have observed that here I wrote tRNA. So this is not the shadow, but probably you haven't noticed that because you were not looking for tRNAs in these slides. And this is true for sequencing. So let's take these northern blots as the ground truths and accept that most, at least these EVs, mostly contain full-length tRNAs and not tRNA halves. When we did small RNA sequencing, we actually sequenced these blue bars over here are tRNA halves, but we were not seeing the full-length tRNAs because the sequencing method that we chose is not capable of actually sequencing full-length tRNAs. So we actually simply ignored their presence. Matthew Shurtleff from Randy Sheckman's lab, they published this paper where they use a thermostable intron-derived reverse transcriptase. The sequencing method is called TigerSeq, and in this case, they mostly sequence full-length tRNAs in the EVs, or almost full-length tRNAs. So depending on the choice of the sequencing method, the output can be completely different. And based on our northern blot results, I will say that this is probably more representative of the truth than standard small RNA sequencing in this case. So let's move on to our second assumption, that microRNAs are abundant inside EVs. So this is quite an old paper from Munesh Tewari's lab. There's also a paper from the same year from Andy Turkinovich and collaborators showing pretty much the same. Here in plasma from different donors, they fractionated plasma using site exclusion chromatography. So now they have the fractions 8, 9, and 10 actually contain the vesicles. Fractions 14 onwards contain proteins and ribonucleoproteins. And they follow by qPCR different microRNAs. And as you can see, LED7A is systematically associated with vesicles, but others like miR16 or miR92A, they are systematically associated with ribonucleoproteins. So they are outside vesicles. And actually they show that they are in complex with argonauts. Then they did this in a more systematic manner and realized that there are a group of microRNAs that tend to be associated with EVs. There are a group of microRNAs that tend to be outside EVs in human plasma. And then there are some microRNAs which are both inside EVs and outside EVs. So inefficient EV purification protocols will not only be problematic in terms of when we want to study tRNA fragments, because we can have contamination with non-vesicular tRNA fragments. It's also problematic with microRNAs, because we can also have contamination with non-vesicular microRNAs, which are also abundant and which are also stable. So this extends to different RNA subtypes. So the bottom line here would be, we need stringent EV purification protocols if we want to study by sequencing the RNA content of EVs. How many microRNA copies are per EV? You are probably aware of this, but we used to think in EVs like this frame over here, full of passengers. So the passengers will be the microRNAs. So at least when I started working in this field, I saw that EVs were full of microRNAs. And when one cell will internalize one EV, then a lot of microRNAs will go into that cell. But then people start measuring that. So they measured microRNA levels by digital PCR and EV particle levels by nanoparticle tracking analysis. And the ratio actually shows that there are very few microRNAs per EV. There are microRNAs per EV, but their number is not high. There's much less than 1 microRNA per vesicle. Actually, on average, there is 1 microRNA copy per 100 vesicles in this study. And we did a similar calculation. And for miR21, which is the microRNA more abundant in our samples, we measure 1 copy per almost 100 vesicles. So the tRNA hubs are more concentrated inside EVs, if they are really inside EVs. And if we look for full-length tRNAs, which we will not able to sequence in this study, they are probably beyond the 1 per vesicle limit. But when we want to study microRNAs, let's bear in mind that each EVs might contain one copy of our microRNA of interest or 0, and probably 0, most of them. So actually, the train, when it comes to microRNAs, will probably look more like this. Our third assumption is that cells select specific microRNAs for release into EVs, presumably to communicate a message. So this relates to the mechanism that cells use to release microRNAs into the exosome space. And again, when we did this small RNA sequencing on large EVs, small EVs, and the non-vesicular fraction purified by ultracentrifugation, we are plotting here the abundance of the microRNAs inside the cells and outside the cells in the extracellular samples, and we see a correlation, a pretty good correlation, meaning that the microRNAs which are more abundant in the cell tend to be those more abundant inside EVs, and the microRNAs which are less abundant inside the cell tend to be less abundant in the EVs, and we also observe this by digital PCR. But you have probably heard the opposite. Many people have observed that the microRNA content of an EV does not resemble the intracellular abundance of microRNAs in a cell. So there's a discrepancy here. One possibility could be related to the way we grew cells. So we use serum-free media, and as I will mention in a while, that can make a huge difference. But I'm not sure that it's really a case of the data. It can also be a case of how we interpret the data. Because I started this is from figure 1 from a paper that was published in Nature a few years ago. And it actually is a paper explaining the mechanisms that cells use for selective microRNA release into the EVs. And in the sample type, the authors observed that 57% of the microRNAs they sequenced had signs of selective distribution. So they were either cell enriched or small extracellular vesicle enriched. But actually, if we go to the supplementary materials of the same paper, and we plot the normalized expression in the cells and the normalized expression in the Vs, something very similar as we did here, actually we can see each dot is a microRNA, that the data is highly correlative. So microRNAs which are more abundant in the cell tend to be also more abundant in the EVs. And this is data from this paper. There are some microRNAs which are really enriched in the EVs, like these two over here, that they are really above, way above their expected abundance based on their intracellular abundance. But I wouldn't say that half of the microRNAs escape from this correlation, at least looking the data in this way. I mentioned earlier that it was relevant, the fact whether we grew cells in the presence of serum or not. And in 2016, the group of Anna and Krzyzewski, they sequenced fetal bovine serum, they sequenced miRNAs in FBS, but also in EV-depleted FBS, which is what you probably use to grow your cells when you want to purify EVs. The problem is that EV-depleted FBS might deplete EVs better or worse, but it does not deplete non-vesicular microRNAs. And as we observed earlier, there are a lot of microRNAs and tRNA fragments and many other RNAs that are not associated with EVs, so they are not depleted by EV depletion protocols. And actually, when they sequence the microRNAs that are more abundant in vesicle-depleted FBS, then with Ken Whitwer, and actually I stole this slide from Ken, we wrote this review, and actually it's pretty clear that many or most of the microRNAs that in the literature are routinely said as being EV-enriched tend to be those that are more abundant in vesicle-depleted FBS. So a clear example of this is microRNA 122, because this is highly expressed by hepatocytes, and it's quite liver-specific. It's abundant in the blood because liver cells release it into the blood, but it's typically not expressed by cell. And it appears as an EV-enriched microRNA in many studies involving the use of FBS. But FBS is not the only source of potentially contaminant microRNAs. And I really like this study that was published in the Journal of Extracellular Vesicles in 2019. Here, the authors were interested in studying intercellular communication between neurons and astrocytes. So they purified EVs and they did small RNA sequencing, and they have this volcano plot. So here is the log fold change between cells and EVs. So cells over EVs. So here we will have the microRNAs that are enriched in the EVs respect to cells. And as you can see here and here, microRNA 122 and microRNA 451A are extremely enriched in the EVs. And they could move forward to try to establish the role of these microRNAs in the communication between neurons and astrocytes. But they realized that these microRNAs are actually among this list of usual suspects of microRNAs that tend to be contaminants of FBS. But the problem here is that they grew cells without FBS. They use a serum-free defining media formulation. So if these microRNAs were contaminants, where was this contamination coming from? And they actually tracked that to the catalase enzyme that this defining media formulation had. So this media, it was a commercial media, had some recombinant proteins, some purified proteins, and one of the purified proteins was catalase. And catalase can be obtained from two sources. There are two main sources for that, which is bovine liver and bovine erythrocytes. And then they went to vendors that sold catalase, either coming from bovine liver or bovine erythrocytes, and they found that when the catalyst was coming from bovine liver, it contains a lot of miR-122, which as I said, is highly liver specific. When the catalyst was coming from bovine erythrocytes, they contain a lot of miR-451, which is highly enriched in red blood cells. And if they use catalase coming from yeast, it didn't contain none of these microRNAs. So they repeated these experiments and these microRNAs were gone. So they were really contaminants. And the contamination here was coming from a protein. So the sources of contamination, of small RNA contamination, are multiple. And this is something that we really need to consider. So last, let me check okay, they're fine. So we also think that once inside cells, once EVs release the microRNAs into cells, these microRNAs will efficiently silence their target mRNAs. But again, the fusion events between EVs and the plasma membrane might happen, but what seems to be the case in most of the cases is that EVs are internalized by endocytosis, and the microRNAs or the RNA inside EVs, they need to escape from the endosomes if they want to target a specific messenger RNA. And several studies have tried to address the efficiency of this process where the membrane of the EVs need to fuse with the membrane of the endosomes. Otherwise, the microRNAs or the RNA content of the EVs will end up in lysosomes. And it turns out that this can happen, but it's not an efficient process. So only a small percentage of the RNAs that are internalized by a cell can actually engage with target mRNAs if we are talking about microRNAs. And so this is so I started this paragraph from a review by David Bartel, that is a well-known researcher in the microRNA field. And he's mentioned this, I think this is a criticism that to this field of intercellular communication that is valid, and we need to take it seriously. It says, moreover, the high effective target abundances explain why lowly expressed microRNAs have negligible effects, because a microRNA can target multiple mRNAs. It's not only the mRNA that we want to target in our paper. So there are a lot of potential targets. So if you only have one microRNA, the effect of that microRNA in the gene expression of a given mRNA is limited, but also it needs to compete with a lot of potential mRNAs that can also engage that microRNAs. So actually, we need high microRNA concentrations to have a measurable effect. And according to David Bartel, this in turn seriously challenges the idea that the microRNAs delivered by exosomes might reach consequential levels in recipient cells. So we have vesicles that contain low microRNA quantities. How many vesicles need a cell? Does a cell need to internalize? Considering that the endosomal escape is a rate-limiting step, and most of the RNA content will end up in lysosome, how many vesicles are needed to be internalized in order to have a measurable effect on gene expression? That's something that numbers not really add up when it comes to it might happen in certain circumstances, but it might not be as universal a mechanism as we think. So microRNA content of EVs is low. The selective microRNA loading in EVs might work for some microRNAs under certain circumstances, but might not be always the case. EV microRNA release into the cytosol of recipient cells is an inefficient process. It happens, but with a low efficiency. The effect of a single microRNA on target mRNA translation or stability is also low. So we can say to this point, do we have a communication problem? And I don't think so. I think that this happens in certain cases that we can discuss. But also, RNA-mediated intercellular communication might go way beyond this simple donor or sender and recipient cell mechanism. So we need to think in other ways of intercellular communication mediated by RNA. And in the last 3 minutes, I will go briefly into this. One possibility is that we might have communication without selective RNA release. So this way of thinking in communication where I want to send a message to you and I want to encrypt a specific message, this message in a bottle that I showed earlier, it's not compatible in the way we communicate these days. So this day, I will just open Twitter or Facebook or Instagram and say, take a selfie and upload it to the internet. That's a way of communication because I'm communicating something about me, but I'm not thinking in sending a specific message to anyone. And cells might simply be releasing the RNAs they have inside, which are more abundant. They get to the EVs easier. And that doesn't mean that other cells can actually read and interpret that message. So we might have intercellular communication, even if cells are just passively or non-selectively releasing RNA into the extracellular space. The second thing is that we don't really need endosomal escape to have RNA-mediated intercellular communication. We have RNA sensors in the endosomes. So if vesicular RNA or non-vesicular RNA can actually escape from the endosomal membrane and they can engage in these RNA sensors, we can have a response in the cell, and we don't need the RNA to escape from the endosome to have a response. So we also need to look more into RNA sensors, in endosomal RNA sensors, to understand cellular responses to extracellular RNA. And finally, we also need to update our picture of the RNA content of EVs. So I like this image from a recent review because they are actually drawing vesicles with tRNAs. So full-length tRNAs are an important RNA component of EVs. There are thousands of papers on the effects of microRNAs, of vesicular and microRNAs on recipient cells, but we are not really studying the effects of tRNAs, of full-length tRNAs or other RNA pol 3 transcripts on recipient cells. And we also have free RNA, non-vesicular RNA, which can be inside lipoproteins, inside ribonucleoproteins, but also free. So naked RNAs, some forms of naked RNAs like nicked RNAs are stable enough, and they can also mediate signaling events in an intercellular fashion eventually. And that's, again, a new direction to look at. So this being said, thank you a lot for listening. Sorry for my English limitations. I hope you will have at least grasped the idea of what I tried to, of the messages I tried to convey. And also a huge thanks to our wonderful lab mates, because I showed some results from our lab, and most of them were done by the incredible people I have the luck of working with. So thank you.
Thank you so much, Juan Pablo, for the very nice talk, interesting. Okay, now the questions are open, so if someone have a question, please go ahead. Or you can write in the chat if you want.
I can see that there are some questions in the chat box. Should I read them, or do you prefer to read them?
Yes, because I don't see anyone.
Yeah, so I can see one from Tom Dedrons. Hi, Tom.
Yeah, yeah.
Says, do you want to read, or should I?
Yes, I will read. So
Okay.
Tom, say great talk about naked tRNAs. Do you know why the RNAs preferentially cuts in the anticodon loop, and why not in the two other loops in the tRNA?
Yeah, thank you. That's a great question, and I think the answer, it comes to the structure of a tRNA. So we usually think in tRNAs, thinking in the 2D clover leaf structure, but actually in 3D, tRNAs are more like an L, and the T loop and the D loop actually interact with each other. So actually they are forming a double-stranded RNA-like structure, while the anticodon loop, that's always single-stranded, and for a good reason. The function of the tRNA is to have that single-stranded region to interact with mRNAs. So that, I will say, the anticodon loop is the most exposed single-stranded region of a tRNA, and that's why we think that extracellular RNAs is preferentially cleaved there. But this being said, we also observe cleavage events in the D and the T loop, and we are actually writing in the process of writing a manuscript on that. That depends a lot on the conditions in which the divalent ions in the media, for example. But the preferential cleavage site is the anticodon loop, and I think that's the reason.
Thanks. Mona Batic, do you want to ask directly or by the chat? Mona,
Yeah,
are
hi,
you here?
I can say that. Yeah. So really good talk, really interesting. So I was just wondering, what about other longer RNAs like circular RNAs or mRNAs? Have you looked at those?
Yeah, that's a great question. So regarding mRNAs, we haven't looked at them specifically, but there are some reports that suggest that mRNAs are present in the EVs. They might also be present outside EVs, but they're probably in fragmented form. But inside EVs, well, I have seen some unpublished data from other groups, so I cannot comment, but it will suggest that there might be full-length mRNAs in EVs. Or the mRNAs might be fragmented, but there are mRNAs in EVs, and also people have seen that there's a correlation between the size of the mRNA and the presence in EVs, meaning that very large mRNAs might have a difficult time in getting inside EVs. So that we also need to think in the biophysics of an mRNA and the size it will occupy. And regarding circular RNA, I think that circular RNAs are very cool. Why they are so highly enriched in the EVs, I don't know, but I can really think on why they could be enriched outside EVs in the non-vesicular space. We haven't looked at that specifically, and I don't know if someone has actually tried to see whether extracellular circular RNAs are really inside EVs versus outside. But if we have non-vesicular circular RNAs outside, because circular RNAs are a circle, they are resistant to exonucleases. If they have proteins, if they are interacting with proteins, they might also be resistant to endonucleases. So that will give again an explanation of survivorship bias. So maybe circular RNAs are very abundant in the extracellular space because they are more resistant to degradation, and that's why they get enriched. But this could explain their presence outside EVs. And then the question is their presence inside EVs a contamination with non-vesicular circular RNAs, or are they also circular RNAs inside EVs? So I think all these are the kind of research that I would like to stimulate with this talk. And for that, we need very good EV purification methods, because we really need to get rid of the non-vesicular RNA. And we also need, of course, good RNA sequencing methods, but also northern blots and other techniques, which are old-fashioned, but they might be less biased for some of these sequences.
Yeah, that's good. Yeah, thank you. I mean, other thing, of course, being devil's advocate, when we say non-vesicular RNA, it could also be during isolations, some vesicles break and they release their RNA and it become non-vesicular, but actually cell pack them into an EV, right? So,
absolutely. Yeah.
so there is always error on both ends. So you can't really pick one on the other. But I agree, we need better techniques, and maybe imaging is a solution too. If we can actually see what is inside every EV rather than doing population studies, that will help too.
Yeah, I completely agree with that.
Okay, Chunyu, are you here? Do you want to ask directly? No? So Chunyu ask, have you ever looked at mRNA and the mechanisms that how mRNA are fragmented and enriched in EVs?
Okay, so I kind of tangentially answered this question earlier, but I think another thing that I think it's interesting is that, again, there's a paper from 2017 from Anna Krzyzewski and colleagues, where they looked at mRNAs inside EVs, and they observed that there was an enrichment in UTRs, so 5' and 3' UTR. And again, this could be a consequence of stability. So it could be that cells are packaging UTRs of different mRNAs because they want to release the UTRs, but maybe the cells are packaging the full-length mRNAs, and then there is some RNA degradation inside EVs. So EVs might also package some RNases, or it can be degradation during handling. That's also a possibility. And then the UTRs, because they're way more structured than the coding domain sequence. So that's why they can also be enriched. So I think that when it comes to extracellular RNA, we should always think about RNA stability. So RNAs that are more stable, they might be enriched because of this survivorship bias. But that's actually not, that doesn't mean that that biology is not involved. Possibly the RNAs that are more resistant to degradation are those that might have more chances of having signaling functions.
Hey, thanks, Jonis. Thanks for the amazing talk. As EV uptake is something that happened at a very low rate, less than 1%. What do you think that means in terms of the effect of microRNAs and tRNAs action on recipient cells?
Yeah, thank you. So I tried to give a brief opinion because it's nothing more than an opinion. That yeah, I think we need to see whether we should expect a very rare microRNA in a cell. The cell is releasing EVs to the circulation, and in a very distant organ, a cell is taking up EVs, and then most of the internalized EVs will end up in the lysosome, and only a few will be able to release the microRNAs into the cytosol. And then we need high loads of microRNA concentrations to have measurable effects. So it's difficult to think that we can have intercellular communication in those circumstances. So I think that when cells are really close one to another, so one cell can actually take up a ton of EVs from another cell, then it doesn't matter whether cells have 1 copy per 100 EVs, they are internalizing thousands. And if the microRNA is highly abundant, then it might be also highly abundant in EVs. And then it's at least easier to think about a paracrine effect. But I think we cannot really extrapolate that to every case. But there are other ways where we can have communication. For example, as I mentioned, TLR signaling. So we have TLR7 and 8, which recognize single-stranded RNA. And there are many reports showing that RNA inside vesicles can be sensed by these sensors. And therefore, the recipient cell can actually read a message and respond to it without the need of having actually endosomal escape, which seems to be the rate limiting step when it comes to RNA exchange. So I think that there might be examples of paracrine signaling mechanisms involving microRNAs. Why not? And I think that there are some very good examples in the literature, but we also need to accept that not every EV that we find in plasma contains a message which is going to be to have not any microRNA in an EV in plasma will have a functional effect in a given cell. And there might be other functions involving more abundant RNAs in EVs, like tRNAs, for example.
Thanks for the answer. And Alexis, do you have the last question?
Thank you, Gustavo. First of all, thank you, Dr. Tosar. It was an amazing talk. I love the references. It was poetry, EV poetry. Following the tRNA thread, I would like to ask you about corona proteins. People is talking about corona proteins, right? Like surrounding proteins around extracellular vesicles. So my question is, will it be possible to have tRNAs anchored on these corona proteins? It means tRNAs that is not completely outside or whether inside extracellular vesicles?
I think this is a great point. And let me say that, for example, there's a lot of enthusiasm right now with RNA on the surface of cells, and people have described glycosylated RNA on the surface of cell, and the same would apply. So if the RNA on the surface just an artifact because some cells release the RNA and it's sticky and it sticks to the surface of the cell and it will stick to the surface of the EVs, or is that actually the way how EVs work? And for example, when I mentioned that we use optical gradients to separate the EVs from the non-vesicular RNA, and actually we saw that the full-length RNAs were in the EVs and the nicked RNAs or the tRNA fragments were outside EVs, I think that's good from a characterization point of view, but maybe by doing this separation, we are breaking apart this non-covalent link between non-vesicular RNA and the vesicles, and maybe that's the way in which EVs are, and that's what a cell will see. So I think that's an open question. It's a more refined question because we need to first understand what's inside EVs and what's outside EVs, and then say, in the biological samples, what is an EV? So an EV is not only its internal components, it's also the protein corona, and why not the possibility of having an RNA corona composed of extracellular RNA. So I think that that's an excellent point.
Thanks so much.
Thanks. And the other one is from D. Dimimok. Do you want to ask directly? No? Okay, so he said, or great talk, thank you. And do you think, okay, do you think the focus on EV-RNAs in intracellular communication might miss some of the communication done via non-EV exRNA? Lots of purification method filter microvesicle, apoptotic bodies, etc. And I have seen some proposed pathway for naked RNA proteins bound RNA uptake to which may all be communicating with other cells. Yes.
Absolutely. I think that non-vesicular RNA and naked RNA are kind of the Cinderella of extracellular RNA. And actually, we have been trying to establish whether non-vesicular RNAs and naked RNAs in particular can be internalized by cells and can be sensed by cells, and whether they can escape from endosomes. And well, I don't have a slide for this because I was not thinking that this talk will go in that direction. But we have a manuscript that we plan to upload to bioRxiv in probably next week about this. And the short answer is yes. Non-vesicular RNA and naked RNA in particular is highly bioactive in cells. There are some methodological tricks to see that, because again, it gets degraded in the extracellular space by extracellular RNAs. So you need to inhibit the RNAs to see the effects. And when we do that, yes, they're highly active.
Okay, thank you so much, Juan Pablo. If other one have a question, please ask now. Okay, no, anyone more? Okay, so thank you so much, Juan Pablo, for this very nice talk and this RNAs teaching in EVs. Okay, so also for the assistant, please check our social media for the next talks, and see you in the next time. And again, thank you, Juan Pablo, for everything.
Thank you again for the invitation, and I really enjoyed these discussions of great questions, and so thanks a lot, and thanks everybody for listening. And of course, you can approach me by email or social media for any remaining question that you might have.
Okay, thank you. Bye.
Okay.
Bye, everyone.
Bye-bye.
See you. Take care.